Great, thank you very much for having me. I want to thank Dominique and Bill Gray for inviting me today. I'm Matt Witkowski. I'm from Yannis Ifantis' lab at NYU School of Medicine, which isn't a very popular place to be right now, but here we are. Today I just wanted to describe some of the work we've been doing on the immune microenvironment of B-cell acute lymphoblastic leukemia. And this disease is the most common form of cancer in children. And it's the result of immature B-cell progenitors accumulating in the bone marrow and then disseminating around the body of the individual patient to block normal organ function. And actually to date, BALL is one of the most heavily characterized diseases in terms of genomics and gene expression profiling. Work from Jim Downing's lab at St. Jude and many others was able to show initially that somatic mutations affecting B-cell development were recurrent mutations in BALL. It caused a block in B-cell differentiation in the bone marrow. Then you have another hit that drives transformation of the disease. And that was extremely well characterized back in 2007. Charles Mulligan and many others have then been able to show that recurrent mutations in genes such as Icarus confer poor responses to chemotherapy. And even more recently, bulk RNA-seq profiling, which I've shown here on the right, from St. Jude, described, for example, 2,000 BALL patients. And what you can see here are little dots that represent individual patients at St. Jude. And you can actually stratify each subtype of ALL based on the expression profile quite nicely here. However, despite all this really lovely work describing the intrinsic drivers of B-cell transformation, actually very little is known in the realm of BALL about the extrinsic regulators of leukemic blast survival. So in a recent review that we wrote, and there's been plenty of reviews on the microenvironment of leukemia, a couple that I've referenced below. But really, a lot of the work thus far in the realm of looking at the microenvironment's role in leukemia survival comes from non-hematopoietic compartments, such as, in the case of BALL, adhesive interactions with, for example, the mesenchymal stem cell compartment. Work from Australia was able to show the endosteal osteoblastic niche as a critical driver of ALL survival. But in the case of BALL, actually very little is known about the immune microenvironment within the bone marrow and how this may regulate responsiveness to therapy. And this is of particular importance now because recent developments in ALL therapy are taking advantage of either CAR T-cell therapy or bispecific T-cell engagers to utilize the immune system to drive an anti-tumor response. So really the aim of my study in New York was to really map the immune microenvironment of BALL. Can we actually understand the soil from which this BALL grows and evolves throughout therapy? So here, just to give background on the strategy that we actually used, we took advantage of the fact that ALL is extremely well biobank disease. There are frozen bone marrow mononuclear cells from pediatric patients across the whole range from diagnosis, remission, and relapse at multiple sites. And if we actually look here at what would be found in such a sample, you would have a combination of cells. Predominantly, you would have your BALL blast, but we don't really know about the other cell types that exist within this environment, which we consider the CD19 negative immune microenvironment. And if we look at a flow cytometry representation of a healthy donor, the CD19 positive population is around 10 to 15% in a given patient. But if you look at a leukemic patient, it's around 90% of the cells are BALL. So if you were to take this sample and just throw it into a 10x genomics experiment, you'd really only get data from the leukemic blasts if you were to analyze the sample as is. So we actually took to a modified strategy where we separated by flow cytometry the CD19 positive blasts and the CD19 negative immune microenvironment. And then we artificially enriched for the microenvironment prior to analysis on the single cell RNA-seq platform. So here we have around 5 parts of the CD19 negative group, and we mix in 1 part of the tumor. We completed this for 7 BALL samples, spanning two major subtypes of the disease, the RUNX1 translocated good prognosis form of leukemia and the high-risk BCR-ABL positive BALL subgroup. And we had matched diagnosis and relapse samples from these bone marrow aspirates. And in the case of 6 of these patients, we also had a matched remission sample. And we had 4 healthy controls from the bone marrow to compare this. And these samples all came from NYU, St. Jude, or the Children's Oncology Group. So the first question we wanted to ask is, well, when leukemia develops in the bone marrow, how does it reshape the immune compartment when compared to a healthy individual? So looking at this, we compared our healthy individuals in green and our leukemic samples in blue. And this UMAP representation is a 2D visualization of the transcriptional relatedness of single cells in space. And what you take from this is the closer two of these little dots are together, the more transcriptionally related they would be. And what we found here is already you can observe some clear differences between healthy and diagnosis marrow. Through a series of clustering and gene set enrichment analyses, we were able to identify 6 major subclasses of cell types expected to be in the bone marrow. And this is just verification in a heat map of those cell type identities. But really, the major point of doing this diagnosis healthy comparison was to look at remodeling between these 2 different scenarios. And what we found here is if you look at where the diagnosis samples are enriched, in this bottom orange population where the B cells are, we see here an enrichment of diagnosis B cells. These are actually the progenitors, as we'd expect. You have a block in B cell differentiation. But really the most pronounced change that we observed in the non-B cell lineage was in the myeloid compartment. So that's this purple population where you can see it's predominantly healthy cells. When we look in the non-B cell fraction, we see that in the presence of BALL at diagnosis, there's a significant reduction in the proportion of myeloid cells, suggesting the myeloid compartment is very sensitive to the presence of leukemia in the bone marrow. So we wanted to delve into this a little further. So we just isolated the bone marrow cells from healthy and diagnosis patients. And we were able to identify 6 transcriptionally distinct cell types, spanning from the monocytic and dendritic cell lineages. Just remember, these are mononuclear preps, so you really lack granulocytes in these studies. And when we compared healthy and diagnosis representation of each cluster, we saw at this cluster 5 that the diagnosis patients were enriched for this particular population, which is actually at the apex of this UMAP plot. And these cells do not express CD14, but seem to express transcripts encoding CD16, as well as high levels of transcripts encoding the MCSF receptor, which is important for later on. We also looked in the peripheral blood of BALL patients. Here, we could access 2 BALL patient peripheral blood samples and healthy controls. And what we found here is in the myeloid compartment of these individuals, if you look at this cluster 2, which is orange, we saw an enrichment of this particular cluster in leukemic blood. And once again, this was CD16 positive, MCSF receptor high, CD14 low. And this kind of pushed us toward thinking, well, these may be CD16 positive non-class monocytes. But one of the major advantages of our study was the fact that we can track the immune microenvironment throughout the course of therapy. So if we look at this particular individual with the BCR-ABL disease, at disease diagnosis, you have around 90% CD19 positive, CD10 high BALL blasts, but following induction chemotherapy, we get a dramatic reduction in this population. As you can observe here, this patient goes into a remission, and then around 3 years later, this patient relapsed with a CD19 positive CD10 disease. And we had these samples analyzed by single RNA-seq and could really track how the environment changed throughout the course of the disease. So we ended up sequencing around 100,000 single cells from 7 patients throughout these 3 stages of disease. And once again, we did our very broad identification of cell types at diagnosis, remission, and relapse. And I've showed that here in these UMAP plots, as well as some major markers of each lineage along the bottom, including this CD10, which is our B-cell progenitor marker, and CD68, which is marking our myeloid compartment here. But for the sake of time, we're focusing here on the myeloid compartment that we saw was so sensitive to ALL presence before. So from the 3 stages of disease, we isolated the myeloid specific cells here, and we identified our 6 major cell types. And if we concentrate on this blue population here, we can see it is low in expression of CD14, but high in expression of the MCSF receptor and CD16. And from these box plots, we can see that at diagnosis and relapse, there is a higher representation of these CD16 positive monocytic-like cells compared to the remission, where we see these seem to have gone away. We've made the claim here that these look like CD16 positive monocytes throughout, but we really needed a way to verify this. We used existing data sets from Rahul Satish's lab, where they looked at human monocytic and dendritic cell subtypes, subpopulation, and what we found is what they described as mono2 CD16 positive monocytes was actually transcriptionally concordant with what we were claiming to be CD16 positive monocytes. So what are these CD16 positive monocytes? These are actually known as non-classical monocytes or patrolling monocytes. And in this nice video, this beautiful video actually here, in green, we see labeling of CX3CR1 positive monocytes, and you can actually see them patrolling vascular endothelial cells. So their normal function is to patrol endothelial cells to detect signs of inflammation. Then upon TLR7 stimulation, they recruit granulocytes to the damaged endothelium, mediating the clearance of the endothelial cell and repair of vascular endothelium. And because of their function in inflammation, they have been implicated in many inflammatory diseases such as atherosclerosis, MS, and rheumatoid arthritis. So what is their function in leukemia? We wanted to know. And first, we wanted to be sure that we were looking at non-classical monocytes, because this is all RNA-seq thus far. You need to verify these things. So we performed a technique called site-seq, which is basically matched single-cell RNA sequencing and surface protein characterization. And if we focus here on these are 4 patients independent of our discovery cohort. If you look here at this blue population up here, these are expressing the mRNA for CD16, and we see with the site-seq protein-based antibody that we do get concordant transcript expression of the CD16 mRNA as well as the protein itself. We continued to perform gene set enrichment analysis from other studies to verify the identity of this monocyte population, and perform trajectory mapping, where we were able to show that CD16 monocytes appeared to be the progeny of CD14 positive classical monocytes, which has been shown before to be the precursor of non-classical monocytes. And probably the most simple and effective way to do this was old-fashioned flow cytometry, where you don't need to do so much fancy things here. And here we're able to show that in healthy individuals, you saw these CD14 positive, CD16 dim classical monocytes, as well as a non-classical population in the bone marrow. However, if you look in BALL bone marrow here, we can see that you've really lost this classical monocyte population at the expense of this CD16 positive non-classical population. We quantified that here with 10 additional BALL patients spanning many cytogenetic subtypes, where we could see that in healthy individuals, you skewed from classical dominated monocytic compartment, and in ALL patients, it was dominated by non-classical monocytes in both the bone marrow and blood. It's one thing to identify these populations by single-cell RNA-seq, that's very nice, but are these of actually any prognostic significance to patient outcome? So to look into this, we actually collaborated with Texas Children's Hospital, where they actually collected absolute monocyte counts at disease presentation for all children that came to their institute with B-cell leukemia. So you can see in these Kaplan-Meier curves, the outcome is pretty good for all these patients because, in fact, it is BALL. They do get treated very well, generally in pediatric ALL. But what we noticed is that if you were to stratify patients on those which presented with a high absolute monocyte count compared to those with the low absolute monocyte count, high monocyte count predicted inferior overall survival from the peripheral blood of these patients. In addition, we went to look at a more high-risk subtype of ALL, which is adult B-cell leukemia. And here we used bulk RNA-seq from St. Jude. Now, using bulk RNA-seq, there is a mixture of cell types in your preparation. So the tool CyberSort can actually predict the abundance of rare immune cell types based on what are deemed contaminant transcripts in your bulk RNA-seq. And if we look at this population of monocytes, where we predicted the abundance of monocytes in adult bone marrow, we saw that adults with a high predicted monocyte population in the marrow fared particularly poorly when compared to monocyte low patients. Now, what is the functional significance of these leukemia-associated monocytes? Thus far, it's a very observational study, and it will largely continue that way. Non-classical monocytes, as I mentioned before, patrol the endothelium to detect inflammation. And when we looked at non-classical monocytes from diagnosis patients or healthy individuals, we actually noticed that there was an upregulation in the transcripts associated with patrolling of the endothelium, such as CX3CR1 and PCAM1. Furthermore, our site-seq analysis was able to confirm that if we observe these two columns on the far left, this is in green, the protein readout, in brown, the mRNA readout. These MCSF receptor high CD16 positive non-classical monocytes express particularly high protein levels of endothelial interacting markers such as PCAM1. So we wanted to generate a way to test the relationship between BALL and its environment, and we generated these human ex vivo vascularized niche devices. And these microfluidics devices allow you to form microvessels using HUVEC cells or human umbilical vein endothelial cells, where you can then infuse BALL into the system and they should traverse these microvessels. This is some imaging of CD31, the vascular endothelial marker, and human BALL cell line ray in this DID purple stain. And we notice when we zoom in on these microvessels, the BALL does in fact occupy within the microvessels themselves and can sort of traverse these particular blood vessels to allow us to test the effect of ALL in a vascularized niche. So the setup here was to see, well, does a BALL microenvironment actually promote differentiation of CD14 monocytes towards CD16 positive cells? So we generated microvessels, and we either left devices without BALL versus devices that are seeded with B-cell leukemia for 2 days to allow some remodeling of the vasculature. Then we infused CD14 positive monocytes, waited a few days, and really stained for vascular diameter of our microvessels and CD16 expression. And if we look in the bottom left here, you can see that a device seeded with CD14 positive monocytes alone, they do seed the microvessels, and they largely express CD14 and not much CD16. You get some expression here and there. But if this device was preceded with a BALL SUP-B15, we now see that these CD14 positive monocytes upregulate CD16 quite readily. And this is significant across the two different leukemic cell lines. One other feature we noticed is that when you seed BALL, we actually had a significant increase in the vascular diameter of the microfluidics device. And this could then be rescued by infusing monocytes into the device, suggesting there may be an interplay between monocytes, the endothelium, and BALL. We have a preclinical model of BALL2 as we wanted to explore the chemoprotective function of monocytes in leukemia development. Here we can transplant GFP positive BCR-ABL driven disease into non-irradiated immunocompetent mice. These home to the bone marrow. This is a vascular endothelial reporter mouse where we see the green cells within the bone marrow. We perform single cell RNA sequencing on bone marrow and peripheral blood of healthy and BALL transplanted mice. And we can see here this brown population is our monocyte dendritic cell population, orange is B cells. If we look at TREML4, a marker of non-classical monocytes, in the bone marrow and peripheral blood of healthy individuals, not a lot of TREML4 expression, but when you transplant BALL into these mice, indeed, you see that there is a significant enrichment of TREML4, a non-classical monocyte marker, which we have verified with flow cytometry to show that there is increased Ly6C low non-classical monocytes. As I mentioned before, CSF1R or the MCSF receptor is highly expressed on these non-classical monocytes, as well as other monocytic progenitors and mature classical monocytes. But we thought using a clinically established approach, or the MCSF receptor blockade, to deplete monocytes, we could see whether it affected in vivo growth of leukemia. So we transplanted BALL into black 6 mice, and then we started either IgG or anti-CSF1R monotherapy. We saw no improvement in the outcome of mice with monotherapy. However, because this is a BCR-ABL-driven version of the disease, we're able to treat with nilotinib to target BCR-ABL. And in the solid black line, we see that nilotinib alone increases the survival of these mice. But if you combine monocyte depletion plus BCR-ABL targeting, we get a significant decrease in the recurrent disease, as well as a significant increase in the survival of mice, suggesting that monocytes may play a role in a chemoprotective role in vivo. So just to conclude, we're now trying to look into the relationship between BALL, monocyte differentiation, and the endothelial compartment. As work from Ralph Adams and others has shown that Notch ligand expression on vascular endothelial cells is extremely important for the transition of Ly6C high classical monocytes toward the Ly6C low non-classical monocytes. Is the remodeling of the vascularized niche by BALL promoting this change in Notch expression, Notch ligand expression, altering monocyte differentiation? Or is it just the injury caused by ALL to the vasculature that causes this recruitment of non-classical monocytes, which ultimately repairs its niche and may form a chemo-resistant hub, which is the endothelial compartment from many other studies. So to conclude, we believe we found that monocytes, particularly non-classical monocytes, play a significant role in ALL outcome, and that BALL itself promotes the emergence of non-classical monocytes. But we're really now trying to venture into the mechanistic basis of this, and is there an interplay between the vasculature, BALL survival, and chemo resistance? So from here in, once we get back in action, we're going to be looking at the role of non-classical monocytes in BALL survival, and use our ex vivo human device to test this interaction more extensively. So just to conclude, I want to thank Yannis Aifantis for supporting me throughout this postdoc. He's been an absolutely amazing supporter of my work from day one. Igor Dolgolev, who is the co-first author on this work, which will be out next week in Cancer Cell, so keep an eye out for that. And Anastasia Tikhonova, who's about to move to Toronto to start her own lab, but she's been a really great support throughout these studies. Bill Carroll, Nikki Evanson, who work at NYU and part of the Children's Oncology Group with myself. Chao Ma for his work on the ex vivo device. Charles Mulligan at St. Jude for patient samples at St. Jude, and Karen Rabin from Texas Children's Hospital and the LLS for supporting my postdoc. Thank you very much.